Okay. Very good morning, my dear friends. This is Dr. PADALA Tirupati, Assistant Professor, Department of Botany, Government Degree College for Women, Jagityal. So, in collaboration with the RBVRR Women's College, Autonomous Hyderabad, and SRR Government Arts and Science College, Karimnagar, we are all going to conduct CPGET online free coaching. Today's eminent resource person, Dr.A.KAVITA Madam Garu, Assistant Professor, Department of Botany, RBVRR Women's College, Autonomous Hyderabad. Madam is going to explain today's class in BRYOPHYTA, Anthoceros. So listen carefully, my dear friends. If you have any doubt during the class, please ask our respected resource person. Madam, please start the session.
Yes, sir. Yes, sir. Thank you so much, sir. Hello, good morning, everybody. This is Kavita. So today I'm going to start with the next genera of the bryophytes, that is Anthoceros. So just I'm start sharing my screen, and this is one. Anthoceros. I hope in the last previous class I have done with the Markantia, so which are commonly called as the liverworts, and whereas this Anthoceros, it is commonly called as a hornwort. So just I write over here. They are called as the hornworts. So what do you mean by hornworts? Can anybody? Hornworts. So that means to say the sporophyte of this thallus, it will be emerging out of the thallus. Whereas in case of Markantia, the sporophyte was embedded within the thallus. But in case of Anthoceros, the sporophyte will be emerging out. It will be coming out of the thallus in the just like a small cylindrical long filamentous structure. It appears just like the horn, horn of the animals. So hence this Anthoceros is referred to as, commonly called as hornworts, H-O-R-N. H-O-R-N, hornworts. Okay, right. So when we look into the classification of this Anthoceros, you can see here it is belonging to the division Anthoceratophyta, class Anthoceratopsida, order Anthoceratales, Anthocerataceae, and the genus Anthoceros. Now you can look into this thallus here. So this is the thallus. And you can look into this. These are the long cylindrical sporophyte. This is called as a sporophyte, which is emerging out of the thallus. Okay, so hence they are referred to as hornworts. So when we talk about the occurrence, so it's a common point all we discussed every time. It is also a terrestrial moss plant, a terrestrial bryophyte growing in the moist soil and also in the shady places. And when we go into the some species of Anthoceros, which are generally found in our India, they are Anthoceros erectus, Anthoceros himalayensis, and Anthoceros cambensis. These are the three important and dominant species of Anthoceros, which are generally found in the Western Himalayas region. Now, so when we go into the general structure, okay, of the Anthoceros thallus, when we talk about the external morphology, thallus is again a thalloid plant body. It is dichotomously branched and the branches are irregular. Even Markantia is also dichotomously branched thallus, but here the lobes are irregular. You can, so when you observe here, this is the margins or the lobes of the thalloid plant body, which is not continuous, it is irregular. Right. And it is dichotomously branched. And also we can see the lobes are wavy margin. The margin is not entire. Okay, you can look into here. The margin is not entire. It is not straight. You can look into, this is a wavy margin of the thallus. So that is the difference. You have to always remember, you have to study the type studies in comparison with one another. Then only the points will be remembered very easily. In Marchantia, the lobes were regular and it is having the entire margin. But here in Anthoceros, the dichotomously branched, it is common feature. But what is the difference here? The lobes are irregular, that is, they are not perfectly arranged, and the margin is wavy margin. And all these lobes, they are arranged in the form of a small rosette-like plant. That means how the rose appears, the rose circular, all the lobes are here, they are in the form of a circular origin. So hence the thallus appears like a rosette plant. And the thallus is a attached thallus. It is attached to the substratum with the help of rhizoids, and inside that is internal part of these thallus going to consist of the most important mucilaginous cavities. Now, so another most important point here, please remember, this is a very important competitive exam question. In this Anthoceros thallus, in these mucilaginous cavities, just now I told you, the internal structure is going to consist of some mucilaginous cavities. In these cavities, the Anthoceros is going to find the presence of colonies of blue-green algal member, that is a cyanophyceae member, of which we call it as nostoc. It is showing the symbiotic association. These nostoc colonies, which is an algae, a blue-green algal member, it is showing the symbiotic association with the Anthoceros. And where they are present, these are present, they are surviving in the mucilaginous cavities in the internal structure of the Anthoceros. So, this mucilage, it helps the nostoc colonies to be motile in the in that mucilaginous cavity. So hence this nostoc, it is supplying the atmospheric nitrogen to the Anthoceros, and in turn this nostoc is going to get the food material from the Anthoceros, the bryophyte. So symbiotic association, every time they'll be focusing on this question. Example, or which cyanobacteria is found in the bryophyte, in the mucilaginous cavity of the Anthoceros. So the example is nostoc colonies. Or the question may be in the other way also, like which genera of bryophyte shows the presence of symbiotic association with blue-green algae? The answer will be Anthoceros. Okay, the vice versa, the other or the two-way question, it may be asked like this. So don't try to miss that question. Now, so when I go into, that is external morphology. Now, when I go into the internal structure of the thallus, the internal structure here, it is going to consist of uniform tissue of parenchymatous cells. So I'll show you here the picture. So this is the anatomy of the thallus plant body. Okay, so we can see here, this is the upper epidermal layer and the lower epidermal layer, which is of continuous, it is of parenchymatous type. It is continuity without any intercellular spaces. And in between these two epidermal layer, you can see this is a mesophyll tissue, or we can call it as a central tissue, which is also of parenchymatous and thin-walled cells. And in between this, you can see these are the large mucilaginous cavities, which is storing the mucilage. And you can observe here, this is a mucilaginous cavity with the nostoc colonies here. These are all the nostoc colonies present in that. So hence it is a symbiotic association. Okay. So the epidermis, it is going to present on both sides, and the cells in the upper region, that is from the upper region of the thallus, consists of the chloroplast. So hence they becomes autotrophic, and each chloroplast is going to consist of a pyrenoid storing the food material. And the thallus is thickest in the middle region, that is as it is accumulating mucilaginous cavities. And the lower epidermis shows the presence of the smooth unicellular rhizoid. So this is also an important point here. So that's why I make it as bold. Whereas in case of Markantia, the rhizoid, we can see the presence of two different types of rhizoids. One is smooth walled rhizoids and another one is tuberculated rhizoids. But in case of Anthoceros, we come across only one type of rhizoid, that is smooth unicellular rhizoids. Okay. Now, so this is the internal structure. Now, when we talk about the growth of the thallus is always grows with the help of a single apical cell in the initial stages, and later on these apical cells will keep on dividing and form the rosette-like thallus. Now, when we talk about the vegetative reproduction, so the vegetative reproduction is carried out in the following methods. One is by the death and decay of the thallus. That is, the thallus, when it is going to die, some of the buds or some of the cells of the thallus, it can have the capacity to regenerate and develop the entire thallus, or with the help of the tubers. Okay, so you can, this is a thallus, this is a diagrammatic representation. So you can see here the margins of these, or in the notches of these lobes, we can see the presence of the tubers arising, through which these tubers, they are the storing, they store a lot of food material, and these tubers, when they detach from the thallus, they will develop into a new thallus. Okay. Now, it can be with the help of the gemmae also, or by the persistent growing apices, that is the tip regions, or is also having the capacity to regenerate and develop a new thallus. Next comes to the, so these are the tubers you can see here. The lobes, the thallus lobe, they develop this tuber which is storing the food material, and when these tubers, when they detach from the main thallus, they will germinate and develop into a new plant. And also here also we can see the formation of gemmae, the gemmae cups, the species here which we call it as glanduloses or formaceae or fusiformes. These are the species of the Anthoceros which are going to develop these stalked gemmae, helping in the formation of a new plant. Persistent apices, that is the apical region, the tip regions of the thallus, when they become dry, so they will undergo dormancy, and later on, by the return of the favorable conditions, they will give rise to a new plant. Now, so when we talk about, now that is regarding the vegetative reproduction. Now, when I go about the sexual reproduction, you can see. So this is the green color mat or everything what you can see at the bottom. That is the thalloid, the adult gametophyte or the adult thalloid. And this is the, you can see these are the emerging long cylindrical structures which are emerging out of this thallus. This is the sporophyte. Long, you can, so here also the difference you can find between Markantia and Anthoceros. Markantia having the embedded sporophyte. The sporophyte will be inside the gametophyte. Whereas, hello, can you hear me, girl? Please, anybody can respond?
Yes, ma'am.
Yeah, okay, fine. Right, so you can see here, this is a long cylindrical sporophytes which are emerging from the Markantia Anthoceros gametophyte. Now, so this is regarding what the vegetative reproduction just now I told you. Now, when you go for the sexual reproduction, Anthoceros is both monoecious as well as dioecious. What is monoecious? Monoecious in the sense both male and the female reproductive organs developing in the same plant body. That is a monoecious condition. Whereas dioecious, both male and female are separate together. Right. In case of Markantia, Markantia is a dioecious. We can see the male thallus is quite different from the female thallus. in case of Anthoceros, and that too in the dioecious, the male plants are, that is the male thallus is smaller when compared to the female. And another important point here is, so in case of monoecious, antheridia, they are produced earlier when compared to the Archegonia. That means in the monoecious thallus, both the sexual organs are developed in the same thallus. When they are developed, first is the antheridia and later comes the Archegonia. Okay. And in this monoecious condition also, the sexual organs are deeply seated in the thallus. That is, at the time of reproduction, they are developed internally inside of the thalloid plant body. Okay, fine. Now I'm speaking about the monoecious condition or the development of the antheridia, that is a male sexual organ. So you can see here, this is the thalloid plant body and this is a male reproductive organ, what we call it as antheridia. They are developed in a small chamber, what we call it as antheridial chamber. And another most important point here is regarding the antheridia. Each thallus, that is each antheridial chamber, can fix only 2 to 4 antheridia, but not more than that. Okay, each antheridial chamber, I'll show you the picture here. So this is an antheridial chamber. In this antheridial chamber, we can see the development of either 2 antheridia or 4, not more than that. 2 to 4 antheridia are developed in each antheridial chamber. Okay, and that too they are embedded inside the thallus, and each antheridium is born with a small stalk. It is a globose structure and they are surrounded by a single layer of thick jacket layer. Again, this is a bryophytic character. Reproductive organs are surrounded by a single layer of jacket cells. Next, antheridia inside going to consist of a mass of androgonial cells, or we call them as androcyte mother cells. These androcyte mother cells will undergo reduction division and results in the formation of the male gamete, which we call it as antherozoids, just like in Markantia. And these antherozoids, they are motile and they are biflagellated. So you can see here, this is the antherozoid biflagellated motile. Okay, from each antheria, you can see here, this is a stalk. Okay, stalked antheridium and a globose head. Inside this globose head of the antheridium, all these is the sporophytic tissue or the androcyte mother cells. These androcyte mother cells will undergo reduction division and produces these male gametes, which we call it as the antherozoids. Okay, so how are these antherozoids? They are spindle-like. You can see here, they are very long. They are spindle-like and biciliate or biflagellate, one and the same. And these, they are motile with the help of the flagella. They can swim in the water and can move from one place to another place. Now, next we can go to the development of the Archegonia, which is the female reproductive organ. So, these Archegonia, they will be developing later when compared to the antheridia in the case of monoecious thallus. And these antheridia, Archegonia, how are they? They are the, this Archegonia, a single Archegonia will be developed in the thallus or embedded in the thallus, which is going to form a globose venter. This is a venter at the bottom and a long neck and the neck canal cells. The neck is going to consist of the neck canal cells and the topmost consists of the 4 cover cells. So you can see here, this is the topmost cover cells. This we call it as the cover cells or the lid cells. And this is a long neck consisting of the neck canal cells. And this is a globose venter. And in this venter, you can see the presence of the female gamete, egg, and this is a venter canal cell. Now, at the time of fertilization, this venter canal cell and these neck canal cells they will degenerate and going to create a long passage, an empty space. And at the same time, the cover cells will also degenerate, creating a passage or opening for the anthrozoids to move inside the venter and fuse with this female gamete, the so-called egg. Okay. Now, so this is regarding the development of the Archegonia and the Antheridia. When we talk about the fertilization here, so when the plant becomes wet, that is during the rainy season, with the help of the dewdrops or the raindrops, the anthrozoids, they are attracted. So with the help of the water, they come towards the Archegonia and they are chemotactically attracted towards the Archegonia, and the anthrozoids will move into the venter, passing through the neck, and they will fuse with the egg and results in the formation of the zygote. And I told you one important point regarding the zygote here. Zygote is the starting cell of sporophytic generation. Remember. And gametes. And one more point here is the right. So sporophyte. So remember, for the next generation, that is from the gametophyte, next generation, whatever is going to be started, sporophyte, that is the zygote, is considered as the starting cell. Okay. Now, so this is the fertilization, how it is going to happen. And this is zygote, whatever is so-called zygote is developed at 2N. This will follows by the sporophyte, the formation of the sporophyte. So how is that forming? So this is the zygote you can see here, which is developed within the thallus. Now this zygote, it will undergo many divisions. It will undergo morphological and changes and results in the formation of the sporophyte. So when we talk about the sporophytic structure here, it is divided into 3 regions, that is foot, seta, and the capsule. So here when we talk about the foot is a basal part of the sporophyte region, which is embedded within the gametophyte, that is the thalloid plant body. And the seta, in case of the second part of the sporophyte, what we call it as seta, it is a very, very small. Whereas in case of Markantia, it is somewhat well developed. But here in the Anthoceros, the seta is very small. The role of foot is what? It helps in absorbing the food material from the gametophyte. And seta, the role of seta is for conduction or for assimilation. But here, seta is not well developed. Okay, you can see here, this is a complete foot. You can see only the seta region by few tissue, by few cells. That's it, not more than that. And the well-developed part of the sporophyte of Anthoceros is the capsule region. So you can see here, this capsule, it is the upper part of the sporophyte, which is long, narrow, and cylindrical. Okay, that's why the name is called as hornwort. Based on this capsule only, it is called as hornwort. It is a long, narrow, and cylindrical. Okay, it doesn't have a distinct seta. So just now what I'm speaking about, it does not have a distinct seta. Only a poor development of seta can be seen. Now, the cells of the capsule here are meristematic, that is lower both. This is another important point in Anthoceros. The lowermost cells or the basal part of the cell